The Application of Melatonin to Stimulate Growth in Mung Beans by Bradley Miller. The planet is highly dependent on agriculture to grow the food necessary for human survival. Fertilizers are a key component in growing all the food we need, but in heavy use, they can pollute groundwater, pollute water sources via runoff, and create harmful levels of acidity in soil. A potential solution to this problem is the addition of new chemicals or hormones to boost plant growth using the same amount of fertilizer, meaning less fertilizer can be used for the same growth. Melatonin is a hormone that could fit as an additive to fertilizer. In a multitude of studies, melatonin has been observed during the early growth phases of some plants, with a possible role in increasing how much the plant grows. This information on melatonin in plants is compelling enough to warrant research into whether it is capable of increasing the amount of growth exhibited by the chosen plant, mung beans. Mung beans have a short sprouting time, low resource requirements, and have been used in similar experiments. The objective of this study was to determine whether the addition of melatonin to sprouting mung bean seeds will significantly increase the amount of growth by mass of them. The research hypothesis was mung beans given a 3 mg melatonin dose during growth will show significantly more growth by mass compared to those given just water. The expectations for this study are that there will be an increase in growth in mung beans given melatonin compared to those given just water because of the evidence of melatonin's role in plant growth. The assumptions made when conducting the study are all mung beans will have the same initial mass, the mineral content of the tap water the seeds are grown in is consistent, the time the seeds spend exposed to light during water exchange will not significantly affect their growth, and the growing conditions for both groups will be identical besides the addition of the supplement, and the chosen growth period will be sufficient for the melatonin to have any of its possible effects. The materials used in this experiment were 40 mung bean seeds, 2 8-ounce mason jars, 2 mason jar sprouting lids, tap water, 10 3 mg melatonin tablets, colored tape, dish soap, a stirrer, and a cardboard box, all of which are pictured in Figure 1. The equipment necessary for the experiment is a digital camera, in my case an iPhone, for documentation purposes, and a digital scale for weighing the mung beans after growth, in my case a simple AMIR digital kitchen scale, as pictured in Figure 2. The necessary facilities for the experiment were a growth area, which was at my house and is pictured in Figure 3, and a data analysis area, which was at my house and at my high school. The procedure for the experiment began with gathering and cleaning all materials to prevent any kind of outside factors from influencing the experiment. One of the jars was marked with a piece of colored tape, and both jars were filled with 100 milliliters of water, as pictured in Figure 4. Next, a 3 mg melatonin supplement was added to the marked jar. 20 beans were added to each jar, and they were placed in the growth area, as pictured in Figure 5. And then the box was placed over the jars for 24 hours, as pictured in Figure 6. After 24 hours, the liquids were poured out of each jar, and steps 2 through 8 were repeated for the next 9 days, the final day of which is shown in Figures 7 and 8. Each group of beans was separated onto paper towels, pictured in Figures 9 and 10. And each bean was weighed on the scale in Figure 11, and the mass was recorded. Finally, the data was analyzed. Table 1 shows the raw data for the masses of the mung bean sprouts grown with or without a melatonin supplement. All measurements are in grams. The leftmost column shows the trial, and the other two show the no supplement group and the 3 mg supplement group. Table 2 is a summative data table for all the raw data in Table 1. Measurements also in grams. It includes measurements of mean, median, mode, standard deviation, and variation, with n representing the number of trials. Figure 12 compares the means of the masses collected from each group to act as a visual aid in understanding the difference between the two sets. The mean mass of the no supplement group is shown in blue, and the mean mass of the 3 mg supplement group is shown in red. Figure 13 contains the raw data from the no supplement group with 3 standard deviation y error bars in order to single out outliers. This set contained no outliers. Similarly, Figure 14 contains the raw data from the 3 mg supplement group with 3 standard deviation y error bars. There are no outliers in this set of data either. An independent one-tailed t-test was utilized for data analysis because the experiment is testing for a significant increase in the 3 mg group when compared to the no supplement group, and the mung bean groups grown in this experiment were independent from one another. The level of significance used was 0.05. The p-value obtained was 0.1736. Because the p-value was greater than the level of significance, the results of the experiment do not show a significant increase in growth by mung beans given a 3 mg melatonin supplement. This warrants a failure to support the alternate hypothesis. The results of this experiment show that the addition of melatonin to the mung beans during growth did not significantly increase their growth compared to the mung beans grown in only water. This is not the result that was expected prior to the experiment. From the data gathered in this study, the conclusion that melatonin does not significantly increase growth in mung bean sprouts is accurate, warranting a rejection of the research hypothesis. The objective of the study was reached, but more comprehensive research is required for a definitive answer to the research question. One variable which may have affected the results of the experiment was the length of the growth period. The allotted 10-day time frame, though used in similar experiments, may not have been enough time for the melatonin to reveal its full effects on the mung beans or enough time for the mung beans to process the melatonin. Another variable which may have affected results is the dosage of melatonin. The chosen 3 mg dosage may have provided an insufficient amount of melatonin molecules for the mung beans to be affected. On the other hand, the chosen 3 mg dose may have provided too much melatonin to the mung beans, which might alter its effectiveness. A number of modifications can be made to the experiment conducted in this study to reach a more definitive conclusion. The experiment conducted in this study took place over a 10-day period, which may have been insufficient time for the melatonin to have its full effects on the mung beans, or for the mung beans to exhibit any effects the melatonin may have had. In further study, the growth period of the mung bean sprouts should be increased. Additionally, the dosage of melatonin used for the non-control group can be adjusted to higher or lower amounts. In further study, different melatonin dosages from those given in this study should be given to the non-control group. Finally, experiments with different plants may yield different results, as different plants have different growth mechanisms. Thank you for viewing my project.